Hi there, everyone. Thank you so much for joining us today. Uh, my name is Elizabeth Errico. I'm the Executive Director of JBRF, and I'm so happy to have with us today one of our board members, Sarah Spray, and our guest speaker, Dr. Herb Lachman, who is a professor of psychiatry and behavioral science at the Albert Einstein College of Medicine. Welcome, Sarah. Welcome, Herb.
Thank you.
Before I turn the floor over to Herb, I am just going to give a quick background on the discovery of the fear of harm diagnosis, just to contextualize how Herb's work relates to our focus at JBRF. So, fear of harm was first identified by researchers back in 2006, and it's quite a revolutionary diagnosis. Because it flips the whole paradigm of how we in the field understand and treat mental illness. It sort of flipped it all on its head. You see, when the powers that be were editing the Diagnostic and Statistical Manual of Mental Disorders in the late 1970s in preparation for the release of the third edition in 1980, they decided to take symptoms and put them into clusters and give those clusters disorder names. And they did this in part so that researchers in different parts of the country could have a common vocabulary to use in order to talk about what they were studying and what they were learning. But the process of this caused the committees to make a very conscious and public decision that they were going to identify these disorders, and these were their words that they published in the DSM, that they were going to be neutral with respect to cause. And what that means is that any potential causes of the disorders that were listed in this book weren't explored or taken into consideration. But in 2006, JBRF researchers were analyzing the data from a massive study of symptoms that were present in kids who had been diagnosed with bipolar disorder. And they saw the symptoms of a new disorder emerging. So this new disorder that they saw emerging out of this data, they called fear of harm because the characteristics of this disorder not only followed the classic symptoms of bipolar disorder, but it also included unusually high levels of anxiety, including separation anxiety and fears that they or someone that they loved would be hurt or killed. It had high levels of defensive and reactive aggression. And there were significant sleep disruptions. Now, the reason why this discovery sort of turns the DSM model on its head is that these symptoms seem to flow from a temperature disruption in the body. This is an objective, measurable physical symptom that seemed to be resulting in behavioral challenges. With the discovery of this, there was the realization that fear of harm symptoms appeared to potentially have a cause that needed to be examined. And this theory bore out as researchers set out to find treatments that would directly affect body temperature. And what they discovered was that treating the body temperature disruptions put many of these other symptoms either into remission or significantly reduced in prevalence. This returned a meaningful quality of life to kids and families who had really been at the mercy of these symptoms for years. So simply put, finding a cause helped lead to effective treatment. But this is not the end of the journey. This is, in fact, just the beginning. And Dr. Lachman is here today to talk about how his work in examining the molecular and genetic basis of bipolar disorder and schizophrenia fits into this larger picture of finding causes for mental illnesses, which could eventually lead to better treatments. So, Herb, take it away.
Yeah. Okay. So I'm going to go to full screen mode to make the slides easier to see. And that's me. And I'm going to make these slides, I'm going to send these slides to Elizabeth along with my email address in case anybody wants to look at them at your leisure. So I'm at the Albert Einstein College of Medicine, and I started my life training in internal medicine and hematology. And I did research on the genetics of leukemia before I switched to psychiatric disorders in the 1990s. So I do wear a lot of hats, as the saying goes. And I become interested in finding genes involved in psychiatric disorders. And when I say finding genes, there are no unique genes in patients with bipolar disorder or schizophrenia. We all have the same genes, but we're looking for are mutations in those genes that affect the protein made by those genes. So when I say finding genes, it's a loose term, meaning we're looking for mutations in those genes. And the reasons for that, I mean, one is that it's extremely interesting to find out how these mutations can cause such profound changes in a person's behavior that can last a lifetime. But there are more practical reasons for doing it, one of which is that many patients don't respond to the medications that we have available. So, for example, in schizophrenia, we have lots of really good medications that can control the psychotic symptoms, but nothing yet to improve the cognitive dysfunction that they have. And there's also a substantial cognitive dysfunction in many patients with bipolar disorder that also is not addressed by the available medications. So there's a lot of room left to find more useful therapies. The medications that we do have, especially the antipsychotics, have many substantial side effects, and it results in a reduction in patient compliance, and it causes secondary problems that are almost as bad as the primary behavioral problem. So finding new medications will help that aspect. And then we have the situation where some patients respond beautifully to certain medications and others don't. So for example, there's a very distinct lithium responsive bipolar disorder, and there are those patients who don't respond to lithium. And geneticists are looking for those genetic factors that can determine who will not respond to these medications. Instead of subjecting somebody who will not respond for genetic reasons to lithium therapy over the course of many, many months and not getting a response, we might be able to predict beforehand which patients will more likely have a good response. And the same thing goes for antidepressants like the SSRIs. Some patients respond beautifully to SSRIs, others don't. Who are those people? What is the genetic background in those patients that might play a role in that? And all psychiatric disorders, all the medications that we use have this spectrum of people who respond or don't respond or who develop side effects. And these have genetic bases in many cases. So those are some of the reasons why we want to find these genes. And we sort of following the modern paradigm for patient care, which is called precision medicine, which is we find the genes that are mutated or the mutations in genes that are responsible for the patient's problem, how that affects the encoded protein, what pathways that protein interacts with, and how that leads to disease, and then maybe design specific therapies based on that pathway. This is an extremely optimistic view of the brain. It's a very reductionistic view of the brain. This is very nice. It does work for certain cancers, and it may work for certain genetic disorders affecting the brain. But it's a little bit pie in the sky-ish. But that's the approach that we're trying to take. And we have these new tools for finding genes involved in psychiatric disorders. And the first one is something we call GWAS, Genome-Wide Association, which emerged in 2006 in another disorder. And before 2006, we had nothing. There were maybe 2 or 3 bona fide genes involved in schizophrenia. And this analysis opened up a whole new avenue of genetic discovery that is ongoing. And this kind of study looks, and I'm not going into great detail, but it looks for common variants. And those common variants are variants that everybody who's listening to this discussion have. They don't cause generally any major problems. They exist as part of normal variation that exists in humanity. But if you inherit 2 or 3 of these genes, then the combination of those plus perhaps an environmental factor can tweak you from a personality trait to a psychiatric disorder. And this is the technique used to identify those. And then we have 2 techniques that are used to identify rare variants that most of us don't have, that have a very, very strong effect on the person by themselves. By themselves, they can cause a problem, whereas in the GWAS common variants, you need combinations of multiple variants to cause disease. Here, this one mutation can do it. And the one I'm going to focus on today, I'm not going to discuss CGH-RA. The one I'm going to focus on today is DNA sequencing, which can be done by analyzing the 1.5% of the human genome that codes for proteins called exons, and that's called exome sequencing. Most mutations that cause disease occur in exons, so it's sort of going where the money is. But there are many, many variants, mutations in genes, outside of genes that also contribute to disease. And those can only be found by doing whole genome sequencing. So these are the 2 sequencing technologies that people are using to identify genetic variants in psychiatric disorders. So I'm going to just give you a couple of examples. Here is a very famous GWAS study published a few years ago that examined many, many smaller GWAS studies. Most GWAS studies are pretty substantial on their own. They involve 1,000 or 2,000 patients and controls. And this was a meta-analysis combining all those studies to find common genes that have common variants within them that can contribute to disease. And these peaks are those regions of the genome exceeding this level of this threshold of positive effect that were found in the combination. When you do a GWAS study, normally you find one of these peaks. But when you combine so many studies, you get many, many. And here we have 108 different loci that have genes that contribute to schizophrenia. And the findings kind of replicated findings in other studies, but they had more impact because of the sample size. And one of the genes that was found was DRD2, which is the dopamine D2 receptor, which was really an important finding because most of the antipsychotic medications used to control the psychotic symptoms are inhibitors of this receptor. So that made a lot of sense. You have these antipsychotics that were found empirically. Now we had genetic evidence that yes, this would be a reasonable thing to use in schizophrenia. And then there are many, many other genes that were found that code for proteins that affect that are found on the surface of neurons. Those are receptors like DRD2 and calcium channels. When we find genes that are on the surface of the membrane, of the cell membrane, we get very excited because those are the most druggable targets. It's not that hard to design a drug that interacts with something that is expressed on the surface of cells, and much harder to design drugs that have to get into the cell and target something within. So these are very exciting findings, and it shows that there are subgroups of patients with schizophrenia who might respond to manipulations of calcium channels and to receptors for glutamate, which is the major excitatory transmitter in the brain. And to me, the most important finding scientifically is the association to what's called the HLA locus. And if anybody here is more familiar with the mouse genome, it's called the MHC locus in mice. This is the locus in the human genome that's important for regulating immune function. They are responsible for handling bacterial infections, viral infections, and mutations or variations within the HLA locus are involved in whether and how you respond to the HIV virus and how you respond to now the SARS-CoV-2 virus. So these are really important proteins involved in the immune response. If you ever need a kidney transplant or some other transplant, this is the locus that has to be analyzed to make sure that the donor and you are compatible. Because if you're not compatible at this locus, you'll reject the tissue. And most importantly, in terms of schizophrenia, is that this is the locus that has variations within it, genetic variations that lead to autoimmune diseases. So most, but not all, autoimmune disorders will have a specific signature within the HLA locus that puts them at a high risk for developing things like rheumatoid arthritis, thyroid inflammation, and so on. So this is a critical finding because it suggests the possibility that a subgroup of patients with schizophrenia have an immune or autoimmune basis for the disease. And that has really, really important implications that I don't have time to go into right now, but I'll be glad to address that in the Q&A or by email. So that's what happens when you do GWAS. You find things that you didn't expect, and you find things that open up new avenues for research and possibly for new therapies. Here's a meta-analysis in bipolar disorder showing fewer loci, but you can see here also there's a locus in the HLA locus also has a hit in bipolar disorder. So it looks like even in bipolar disorder, there may be a subgroup who have an autoimmune or some kind of inflammatory process that leads to their psychiatric problems. And here's a calcium channel, which we like, and here's a potassium channel. So you find these things that are really quite important. Now, most studies, so GWAS is, as I pointed out, one of the ways to find genes. We're focusing primarily on sequencing. And the ability to sequence genes has improved markedly over the last 3 decades. The first human genome was sequenced in the early 2000s, and it was the Human Genome Project, and it utilized a classic DNA sequencing technology called Sanger dideoxy sequencing, which is very nice but very labor intensive. It cost $3 billion and 10 years of research to sequence that first human genome. Now we have these new sequencing technologies called next generation sequencing, where we can do the same analysis for a few $1,000 in a couple of days. So this is a major advance. And this next generation sequencing is the technology that was used to identify SARS-CoV-2 back in December 2019. It took a couple of days for the Chinese researchers to get a sample from patients, sequence them, and identify this new coronavirus. All of the new variants that pop up emerge or are known quite quickly because of next generation sequencing. And this is the technology that we use to find genes involved in mental disorders. So I pointed out initially that there were 2 methods for sequencing. One is exome sequencing, the other one is whole genome sequencing. Exome sequencing, again, focuses on the 1.5% of the genome that contains most disease-causing mutations. And here's just the first study that came out looking using exome sequencing to find genes involved in schizophrenia. And this is not that important, but it just shows you that you get a lot of hits, and number of cases that are needed to get a hit is very, very small compared to the number of cases you need to pick up common variants in the GWAS technology. These studies have been carried out in autism, in ADHD, in bipolar disorder, and other psychiatric disorders, as well as inflammatory bowel disease and other non-psychiatric problems. So the hit rate is quite good. And here's a study that we just finished. This is a study that is now under review at the Scientific Reports. And this is a study I was involved with where we identified genes involved in this condition called PANS, which is pediatric acute onset neuropsychiatric syndrome. And this is a very, very enigmatic disease that some doctors don't even believe exists. And we show that one of the reasons why it's enigmatic is because genes that are involved, we found 11 genes in 21 patients, the genes that are involved separate into two broad categories. So if you're looking for immune factors involved, you might miss those factors that are affecting brain cells. So again, it just points out novel theoretical features of the disease. It shows new research pathways. Just this one table here. This table has, for me, 100 years of research. I don't have 100 years of life, and I don't have 100 years of funding, but I have 100 years of potential research just in one table. So it opens up these avenues for investigation that hopefully will be translated at some point into helping these unfortunate kids, who, by the way, resemble in some aspects kids with FOH. And in fact, I have a couple of patients here who actually have meet criteria for FOH. So there's a little bit of a crossover in psychiatric diagnoses. So using GWAS and using sequencing, while we had only a couple of genes before 2006, now we have almost too many genes, an amazing number of genes. Now, this is a little bit misleading because it's not as if every one of these, any patient who has any one of these genes or combination of genes, has a unique feature of their illness, a unique pathway to their illness. These 500 genes in bipolar converge on, let's say, 5 or 10 common pathways. So you might have, 50 genes that have mutations in different portions of the same pathway. So this is very, very daunting to see 500 genes, but it's much less daunting when we say that these 500 genes separate into, let's say, 5 or 10 functional categories, so that you're not just finding a drug that will treat one person or a person who has one mutation. You might be finding drugs that might affect the pathways that some of these genes converge on. In FOH, we have nothing. There are no genes because we haven't started yet. And the reason for that is that it's very hard to get the funding from the NIH to do a study that's not even in the DSM. So if I wrote a grant, or Dr. Popoulos and I wrote a grant to look for the genetics of FOH, it would not get funded because people on the committee, people who are reviewing this grant, will say, what is FOH? It's not in the DSM. So that's one aspect, is to do more research on FOH, make it more of a public disease, and get it into the next rendition of DSM-6. And most genetic studies start with classic twin family and adoption studies to prove that the disorder has a genetic basis, and we don't have those at all in FOH, except for family studies. For me, that's enough, but for reviewers, it might not be enough. So we're at this limbo state. However, there is a solution, which is that we can do whole genome sequencing or exome sequencing to look for rare variants, and do it with a not too substantial amount of money, using private donor funding, and maybe even families who might cover the sequencing costs. The cost of sequencing is in the same realm as an MRI scan or LASIK surgery. It's not outlandish. And in fact, if you go back to this study was not done with NIH funding. It was done primarily with some leftover funds with collaborators in the EU, and half of these genes were found by a clinical collaborator who was actually getting sequencing done on his clinical patients, and they were paying for it, and he was analyzing the data when I contacted him to join forces. So this can be done on a non-NIH track. And there's another study that was just published by a citizen scientist who has a kid with PANS, and they published some preliminary genetic studies. So you can do this without NIH, but you can't do it without some kind of funding. And that is my spiel. And again, I'll send these slides to Elizabeth along with my contact information, and feel free to email me. I must say that when you do genome sequencing on people who have enigmatic disorders, you get a positive hit in about 1/3 of cases. So you will come up empty more times than not, and the sequencing studies that are done will not pick up common variants. It'll only pick up rare variants, and your kid might have FOH because of a combination of multiple common variants, which will not be detected on the whole genome sequencing analysis. So you can miss it quite easily. The point is that if we analyze enough patients, we'll get some kind of genetic hook that will be informative for the larger population. But for your own kid, you might not find anything.
So Herb, there's another question here in the chat. I'm not sure if you see it. It says, okay, we have had it done with a geneticist, and we did it through one of those mail-in ones. We did find out he has a partial deletion of chromosome 9.
Okay.
Oh, okay.
So
yeah,
that's probably a very, very substantial variant. So I don't know whether your kid has FOH or some other developmental problem, but if you send me the where the deletion is, I can take a look at it and see. Yeah, that's to me would be a positive hit. So that partial deletion might have been identified through what's called a CGH analysis, which looks for copy variations, and that's what that sounds like. So that's, in terms of genetic analysis done on the outside, there's a whole list of things that you do first before you do genome sequencing, and one of them is to look for chromosomal problems, and I suspect that that's how that was found. It's pretty common to see that in schizophrenia and in autism, but less commonly found in bipolar, but they are found on occasion.
We also have a question: is the data collected on the MHC always applicable to HLA?
Yes, so the MHC is just what the mouse geneticists call the HLA, because they were discovered at different times. So when I talk to graduate students who work with mice, I say this is MHC. When I talk to doctors who know about HLA, I mention HLA. So if anybody here is a mouse person, I gave both.
One question that I have is that I know a lot of times parents are encouraged to have, genetic testing done, and parents get back these sheets that sort of have recommendations on what medications are, likely to work, what medications might, not be likely to work, and parents oftentimes wrestle pretty significantly with trying to decide whether or not to go ahead and have that testing done, because they're not sure about the applicability of it to their kids' day-to-day treatment. Okay. And I'm wondering what your thoughts are on that process for someone who's not participating in research. They're just trying to get their child help.
Okay, so that's another angle to the genetics. So one angle is the one I described, which is to find genes involved in these disorders. And sometimes that can be informative in terms of what medications might be better. What you're referring to here is what's called pharmacogenetics, which is looking at genetic variations in the genes that regulate the activity of drugs and their metabolism. So you might take a drug, you and I might take the same drug, but because of genetics, I might metabolize it faster, meaning I might need a higher dose, and you might metabolize it more slowly, which means you would need a lower dose. Or you converting a drug, these enzymes also convert a drug into its active substrate through the same kind of biochemical reactions. That's pharmacogenetics. And there are some companies out there that will push pharmacogenetics to see whether you have genetic profiles that suggest that you might require a different medication, or you might require a higher dose. And this is still an area of controversy. It is not that, I mean, I happen to be a big fan of it because even if the information is not 100%, it can be quite useful, but it has not been sanctioned yet by the APA and other medical groups because it's just not enough data yet. And I can think of the clearest example where I would use this is in the use of this drug called Plavix, which is used as an anti-clotting drug in patients who've had heart attacks. There's a very, very clear connection between that drug and whether it will work or not, and a certain genetic profile. And it's so clear, and yet the American Cardiology Association has not yet sanctioned its use. So I would say that you might get useful information, and you know, anything it might be helpful. And it doesn't mean that the medication you might not be, that's not recommended, might not work. You would try the other ones that might have a higher likelihood of working, and then go back to the one that is said to be, you know, less effective. So yes, I would say you can do that.
Meg, hopefully that answers your question. Sorry, Herb, didn't mean to cut you
No,
off.
it's okay. No, I get passionate about pharmacogenetics.
We have another question.
And one of the reasons I'm really involved in that is that one of the major causes of mortality in the United States is polypharmacy, giving too much medication, and that you can get into problems if you have a genetic profile that gives you drug interactions that you didn't expect. So that's why the more science you put into prescribing, the more happy I am, even though my colleagues don't necessarily agree.
We have another question here. It says, can data collected outside of NIH funding be used to establish data needed for DSM inclusion? If yes, are there specific types of testing that would need to be done? What would we ask our pediatrician or geneticist for? And going along with that, there's a follow-up that says, if someone had private genetic testing done, could it be done through Albert Einstein?
Well, the genetic testing would be done through a genetics company. I can do this technology myself, but we are not sanctioned to give any kind of clinical tests to people. So the recommendation would be to get the tests done at a genetics company, and we'd work out the logistics, and then the data would come to me and my genetics collaborators to figure out whether anything of interest is there. As far as the DSM is concerned, I still think that to get into DSM-6 is going to require more of a clinical angle. That Dmitry still needs to, Dr. Popolo still needs to do studies more firmly establish the connection between thermoregulation and these psychiatric issues, and then that has that's a faster track into DSM than genetic studies. Genetic studies might take 5 to 10 years, and it's going to help later on for sure. But that initially, the genetic studies really are designed for us scientists to give us an angle to do research on the neurons from these kids. I'll give you, for example, I do work on stem cells. I make stem cells from blood cells. I can turn those blood cells into neurons. I can take a patient with autism and make autism neurons in my lab. And I can study them in the lab, which I'm doing, and figure out what's going on inside the cell and try to find pathways that are aberrant in those autistic neurons. And I do the same thing for FOH. So if we had a clear genetic cause of FOH, we can make stem cells on those kids. We can turn them into neurons. We can turn them into other types of brain cells. We can turn them into small structures that resemble a fetal brain. And then we can do molecular biology on those and figure out how those mutations are leading to the clinical situation, and maybe come up with additional medications aside from ketamine that might be effective. So it's really more for scientific purposes at the beginning, with the hope that we can translate that as rapidly as possible into something that is useful for your family, as well as establishing a genetic analysis that might be used if the kids go on to have their own kids. That's another important reason for finding a genetic cause. If your kid has a really truly obvious single gene defect that is leading to FOH and psych problems, that can be diagnosed either prenatally or even pre-implantation down the road. So that's a very important aspect of genetic analysis that I didn't mention.
So it seems as though there's a lot of interest in having genetic testing done. Is there a company that you recommend, that you've worked with, that you think is reputable and has good reports that you would recommend to parents to follow up with?
There are a couple. I can send you the list of the ones that I've had contact with. Centogene is one of them. There's many good genetics companies. So that is, and the more of you who do this with your private doctors, I mean, if I can get access to those data, I mean, I might, if 20 of you do that, I might be able to find something that is really useful. And sometimes I find things that are useful that genetic companies don't find. I have my ways of digging into the data in a more advanced manner than just the bioinformatics analysis that they do. I have my own personal mini processor that I use to evaluate these genes. So if enough of you get the data and you give us permission to look at it, who knows? I mean, just in this one conversation, I might get 10, 20, or 30 samples, and we can maybe make something out of it. And all we need to do is publish that first brief study on a couple of cases where we can make a connection between a gene and the known physiology of FOH to have an impact.
So there's another question here in the chat. It says, my son, now 32, has been taking meds for early onset bipolar 2 since age 8. He took lithium cocktail with Lamictal successfully for years, but developed severe psoriasis. He's ultimately tried almost all other bipolar drug combinations with varying but not much success. He has tremendous anxiety, sleep disorder. Would he be a good candidate for pharmacogenetic testing? He's very compliant, but struggles terribly almost daily.
Okay, so it's interesting that you mentioned psoriasis, because psoriasis has is an autoimmune disease. First of all, we see a lot of autoimmune diseases in PANS, these kids with pediatric acute onset neuropsychiatric problems. And some psoriasis is due to activation of an inflammatory hormone called interleukin-17. So I'm wondering whether attacking the psoriasis using these new biological immune inhibitors might be effective. Whoever did that, if you can email me, maybe we can have a separate conversation. That's a very interesting problem that you have, and I can't answer the clinical pharmacology problems. I'm not, even though I'm an MD, I'm not a psychiatrist. I don't treat psychiatric patients other than what an internist would treat in his or her practice. So I do see some patients with relatively simple psych problems that I will treat. But in a complicated case like this, you need to discuss that with your psychiatrist. And yes, and the pharmacogenetics testing might be helpful. I don't know whether or not, if you're implying that the drugs might have brought on the psoriasis, that's something I don't know. I don't think that's the case. I think that maybe what you have here is an underlying autoimmune disorder, and that's that one manifestation is psoriasis. And now I'm really speculating, okay, that maybe the psychiatric problems might also have some kind of autoimmune basis behind it. But I'd be very interested in talking to you further, and I need to review the psoriasis literature, make sure I gave you the right cytokine. But I think that maybe a biological treatment with these new drugs that are very effective in psoriasis, that inhibit certain arms of the immune system, I'm wondering whether if that might work for the psychiatric problems. That would be incredible.
Yeah, Herb, I'm wondering if you can talk a little bit more about the autoimmune connection, because I know a lot of parents are looking into that now, they've either had or possibly have been considered, the PANS diagnosis. There's a lot of interest right now in a particular protocol by another physician who works with the immune system and inflammation to reduce symptoms. And so I know that's an area that a lot of people have interest in. And I'm wondering if you could talk a little bit more about the intersection between inflammation and autoimmune and bipolar and psychiatric disorders.
Well, that's a lot, but I'll try to distill 20 years of research, 10,000 papers.
Reader's Digest version.
Into a 30-second or 1 minute, yes, the Reader's Digest version. So yes, so first of all, there are two things to think about. One is autoimmune, and the other one is hyperinflammatory. Autoimmune means that you have some kind of antibody that was produced because you have an autoimmune underlying problem, and those antibodies attack brain cells. This is seen in something called anti-NMDA receptor encephalitis. So you get these autoantibodies, they penetrate the brain-blood barrier, they attack glutamate neurons, and you get this really bad encephalitis with psychiatric symptoms that resemble schizophrenia. That's the autoimmune aspect. Then we have a hyperinflammatory aspect. We don't have autoantibodies, but you might have an increased release of these chemicals called cytokines, which can affect the immune system and cause a hyperinflammatory reaction, like psoriasis, like many other, like inflammatory bowel disease. In Crohn's disease, you don't really have not too many kids or people with Crohn's have an autoantibody, but they do have a hyperinflammatory response in the gut to gut bacteria. In the brain, I believe that the microglia in the brain, which are the immune cells of the brain, are hyper-responsive to various insults that will activate the microglia, cause them to produce these cytokines. And those cytokines have an adverse effect on brain function. That's something that we are actively pursuing in my lab. So there's the autoimmune phenomenon. Autoantibodies are being produced that attack parts of the body, like in lupus, or you have a hyperinflammatory problem where you have overactivation of certain arms of the immune system, leading to the production of cytokines and other chemical juices that can have an adverse effect on brain function and cause anxiety, OCD, cognitive functions. You might have heard about cytokines in the context of SARS-CoV-2, the cytokine storm, which is responsible primarily for the inflammatory process that happens in the lungs that leads to death. Those same cytokines cause long COVID. They penetrate the brain-blood barrier, and they affect cognitive function, energy levels, mood, etc. So these cytokines are very potent effectors of brain function. So in the case of the HLA locus in schizophrenia, and in some cases bipolar, we don't know whether we're dealing with an autoimmune disease per se, because we haven't found any autoantibodies yet in those disorders, or a hyperinflammatory disease where you have certain immune cytokines that are adversely affecting brain function and adversely affecting brain development in fetal life. So if a mother is pregnant and has a serious autoimmune disease herself, or she gets exposed to influenza virus, the cytokines that are released during those episodes can penetrate can pass through the placental barrier and adversely affect brain function. So there are many, many arms or aspects to the neuroinflammatory features of these conditions. And we just don't know yet in schizophrenia. I suspect it's more of a hyperinflammatory problem as opposed to a true autoimmune problem. Did that sort of answer the question?
It does. And we have a comment here from a mother talking about her child who has been diagnosed with FOH and is in treatment for FOH, which are helping, but has continued inflammation, which manifests in mood symptoms and migraines. And they are beginning a anti-inflammatory protocol to reduce cytokines. Is there any additional research you might point parents towards in this area that they could look at?
Now, when you say here's I see the word anti-inflammatory protocol. That is, that sends shivers up my spine. Okay, because yes, there are true anti-inflammatories, non-steroidal anti-inflammatories, prednisone, and some of these immune modulators that are now being used in inflammatory bowel disease and psoriasis. Those are medically sanctioned anti-inflammatory protocols. But I suspect that you might have gone to a doctor who sort of has their own unique anti-inflammatory protocol that might involve dietary changes and things like that. You're on your own there. I am a little bit suspicious about that. I'm open to data that can prove me wrong. If you can show me that going carb-free and gluten-free truly has a positive effect on autism and other conditions, show me those data, and I'm happy to change my mind. But I suspect that you're seeing somebody who has their own unique anti-inflammatory protocol.
Yes. Kyle, you mentioned the specific protocol, and yes, we do know which protocol it is. We're just not advertising it.
Oh,
per
okay,
se.
okay. I know you guys are desperate. Believe me, I know that you have these sick kids, they're not responding well or whatever, and you're seeking out some kind of treatment. So whatever you're doing is not wrong. I'm not, I would do the same thing if my kid had these problems. So you're doing, you can only, I mean, I totally understand why it is you'd seek out some of these people, but just be cautious that these people are promoting their thing online. They're not promoting it in PubMed. You don't see those papers on PubMed. You're seeing it online. So they're promoting their own agenda. And God bless them. If they're right and they're onto something, publish those data and let people like me know. I would love to have a simple solution to these complicated problems. But I'm a little bit I'm a skeptic. But I understand exactly why you would be why you do that. All of my parents' families, they all go to these doctors too, and some of them really swear by the treatments. And again, how much of that is wishful thinking? This is why we do double-blind experiments. We do double-blind experiments because the placebo effect is so gigantic. I mean, it's unbelievable how big it is in psychiatric disorders. These have to be done double-blind, but in the meantime, you can't wait 20 years to do a double-blind study on some of these treatments. As long as it's not harmful, except to your pocketbook, then by all means, go ahead. But I'm a skeptic.
So we have another question here. It says, is there a way that cytokines can be modified and used to cross the blood-brain barrier for healing the brain, like with cancer gene therapies?
Okay, so that's a very good question. And yeah, there are cytokines that have both pro and anti-inflammatory properties. And most of them don't penetrate the brain-blood barrier unless the brain-blood barrier is compromised, as it is with infections and certain inflammatory disorders. And yes, so we are looking actually for those cytokines and something called chemokines to see which ones might have anti-inflammatory properties that might reduce brain inflammation. We are at the very beginning of this research, so I can't make any promises or guarantees. But yes, in theory, if you can identify that subgroup of cytokines and chemokines and other inflammatory modulators that can penetrate the brain-blood barrier and have an anti-inflammatory effect, this is why we're doing this research. As far as specific inhibitors of these cytokines, the ones that have pro-inflammatory activity, there are indeed specific inhibitors to things like TNF alpha, to interleukin 17. So these are very legitimate drug targets. And you can't, if somebody has, let's say, an excess of interleukin 17 that's crossing the brain-blood barrier and causing brain inflammation, we have these monoclonal antibodies that will block interleukin 17. So in theory, yes, you can modulate these. You can modulate these, and they are being modulated for other inflammatory disorders, and increasingly, it will be used in psychiatry.
Herb, earlier you mentioned the idea of
By the way, this is a great chat message. We'll take placebo and voodoo. God bless you. Yes, so would I.
I think we all agree with that. So earlier you talked about being able to use stem cells to grow brain cells and how do you collect those stem cells? If families wanted to be able to do that kind of
Okay,
research,
so we're
how
talking
do you
about two different
collect
kinds of
it?
stem cells. The stem cells you're talking about are mesenchymal stem cells or cord blood stem cells that are used or being considered for use in various neurodegenerative disorders. And you can collect those from cord blood if you have the foresight and the money to store them. You can collect them from your own mesenchymal cells from fat and bone marrow. That's not the stem cells that I'm talking about. Those are stem cells that can be converted into cartilage or neurons, in theory, to treat Parkinson's disease, or in my case, bad cartilage in my knees. Those are those are studies that are still being investigated. The stem cells I'm talking about are stem cells that resemble embryonic stem cells. They're called induced pluripotent stem cells, or iPSCs, and we take blood cells, regular blood cells, and we can convert them into stem cells using this technique that won a 2012 Nobel Prize for this Japanese investigator, Shinya Yamanaka. And we can take those stem cells and they behave in culture the way embryonic stem cells behave in utero. They can be converted into blood cells, bone cells, gut, kidney, and brain if we treat them with the right juices, with the right hormones and growth factors. So we're making stem cells to model these conditions. We can't take brain cells from a kid with autism, but we can make those brain cells from blood cells. So those are two different kinds of stem cells that I think there's some confusion there about the mesenchymal, which is used for regenerative medicine, versus my use, which is to model these conditions in the laboratory.
So the stem cells you use are taken from a simple blood draw?
Yeah, well, I make them. We take white blood cells, and using this 2012 Nobel Prize discovery, we can convert those blood cells into stem cells. And those stem cells we can convert into neurons and three-dimensional structures that resemble the first trimester fetal brain. And I'm not the only. There are many researchers that are using this technology to study these conditions. And we're not using them to regenerate the brain, although some people are using them to make neurons that might replenish damaged dopamine neurons in patients with Parkinson's disease. People are using those stem cells, these induced stem cells, for regenerative medicine purposes. But for me, I'm using it to grow autism neurons and bipolar neurons in the laboratory.
Well, we are just about out of time. I want to respect everyone's schedules and the time you took out of them to be here with us today. So we'll wrap up now. What I will do is in combination with Herb, I will put together a summary of today's talk and put in some of the resources that we talked about, including, it looks like Herb, you put your email address in as your screen name.
Oh, great.
If anyone wants to reach out directly, feel free. Or you can, if you don't have a pen and pencil right now to write it down, you can reach out to me and I can connect you to Dr. Lachman. And if anybody would be interested in maybe participating in a study of some kind regarding Dr. Lachman's work, please let me know because we would be happy to coordinate and facilitate you getting the genetics information to him. Yes, Kyle.
Hi, everybody. I just wanted to let everybody know who's on here who might be interested that I started an FOH support group on Facebook, and we have a small but growing contingent of parents from all over the country who are always looking for support, as I'm sure people can imagine.
Right.
Yeah, and of course,
and Elizabeth has that information.
Yeah, and of course, JBRF also has an online support group that meets 3 times a week for parents. We also have a private Facebook group just for parents of kids who have fear of harm, where we share information. I know there's some families who are in both our group as well as Kayo's. It would be great for us to coordinate that so that we can share across groups. And of course, all of this work that we do is made possible by the generosity of our donors and the people in our community who support our work. We have just finished our year-end campaign. We will have our spring campaign in May, which is focused around Children's Mental Health Awareness Month. And at any time, you can go to our website, jbrf.org/donate, and make a contribution on our safe and secure online platform. You can make that a one-time donation or a recurring monthly donation, which allows you to make consistent contributions that allow us the flexibility to be able to seize opportunities when they come along and to be able to predict our financial possibilities. So we are deeply grateful to everyone in our community for being a part of what we do and helping through research and education and outreach to improve the lives of children and families who live with bipolar and fear of harm. So thank you. And we really appreciate you being here today. Again, if you have any questions or need anything, please don't hesitate to reach out.
Nice meeting you all, and thanks for the provocative questions.
Thank you, everyone.
Thank you.